Thank you, Steve, for the kind introduction. And thank you very much to the organisers for asking me to come and talk here. As some of you may know, I started off doing my PhD with Venki Ramakrishnan on the ribosome, and then I moved over to Ron Vale to work on dynein, which I've worked on ever since. So this is sort of my home crowd, as it were. Ribosomes, motor proteins, this is the best it gets. So what I want to do today is to tell you about mainly our structural and functional studies on dynein. And many of you may know about this microtubule motor, but by way of introduction, I'd like to show you a short clip from a BBC documentary called The Hidden Life of the Cell. So this documentary was actually mostly about how viruses infect cells, but dynein played sort of an important bit part in this film. And so here we are, dynein binds to microtubules, binds to ATP, walks along the microtubules, and I always think there must be some sort of cheesy music playing at this point of the movie. So, and its role, of course, is to drag cargoes. Here is this virus, and it's taking it off into the sunset of the cell, which is, of course, the nucleus, where that virus is going to replicate. Okay, so as Steve said, there's only one dynein, and it's responsible for all of the transport towards the minus ends of microtubules. And it really does carry an awful lot of different things. There are all these different types of membrane vesicles, there's a whole nuclei, RNAs, different proteins, and it also has a whole load of roles in mitosis. So it's found out at the cortex, where it helps to position the spindle. It's found at the spindle pole, where it's important for forming that structure. And it's also found at the kinetochore, which is where it's monitoring the attachment of microtubules. And again, as Steve said, there are masses of different kinesins. Those are the plus-end-directed motors. There's about 29 of them required to do the equivalent functions. And so how is it that dynein manages to do so many different things with just this one motor? I should add, of course, there are other dyneins, and most of those that are involved in the axonemal structures. So because dynein is perhaps involved with lots of things, there's also lots of roles in disease. And some of the classic examples of this is viral transport, particularly neuronal viruses like herpes and rabies, which have to infect the neurons and travel all the way back down the axon into the cell body. They absolutely need dynein. But actually, if you look through the literature, it seems almost every class of virus uses dynein at some point of its life cycle. Sometimes it's to assemble components ready for new viruses to be made. Sometimes it's assembling viruses in a compartment where they can replicate. There are all sorts of different functions. Sometimes it's just rearranging the mitochondria in the cell in order to allow proper viral synthesis. Another set of diseases are caused by mutations in the dynein gene. So this is a wild-type mouse. When you pick it up by its tail, it hangs its legs out. But when you take this legs at odd angles mouse and pick it up, it can't do that because of motor neuron degeneration. And this mouse is caused by a single point mutation in the dynein heavy chain gene that causes this phenotype. And as they're increasingly sequencing different human patients, they're finding similar mutations all over the dynein gene, which cause this degeneration. And the other example then of mutations in dynein are involved with neurodevelopment. And this is the sort of first example, and perhaps the most extreme, where this regulator, LIS1, which Steve has done a lot of work on, is mutated, and the neurons fail to migrate properly as the brain develops. So you get this sort of smooth structure as compared to this indented structure in a normal brain. And again, mutations throughout dynein are causing all sorts of different levels of neuronal defect, from some quite mild ones to some very severe ones like this. So let me introduce you to the basic components then of dynein. It's made of 2 copies of this heavy chain, which is really quite large, over 4,000 amino acids long. And at the C-terminus, you've got this motor domain. That's what actually moves along the microtubule. And then at the N-terminus, you've got all these other accessory chains bound. And this structure forms the tail. This is where cargoes interact with. So dynein doesn't work on its own. There's an essential cofactor called dynactin. This contains another 23 different subunits and is about a megadalton in size. So when you put this dynein and dynactin together, you've got a structure that's 2.4 megadaltons. It's about the same size as a ribosome and the same sort of complexity in order to do this transport process. Okay, so when I started my lab, I was a crystallographer and I was working on the motor domain. But what I want to tell you about today is how my lab got into Cryo-EM, which, as many of you will know, has undergone a revolution recently in terms of what it can do. And once I've done that, I'll tell you about how we got into that by doing the dynactin structure and how it interacts with this tail. And then in the second part of the talk, I'll go into how that's regulated and how this dynein-dynactin machine binds different cargoes. So let's look at dynactin first. This is what the model was of how dynactin looks like before we started the structural work. And the fascinating thing about it is that it's built around a short filament that's very similar to actin. It's made of an actin-related protein 1. And so the big question is, why do you have an actin-like cofactor for a microtubule motor? And I think, well, I'll try and explain that. So we thought very early about trying to do some structural work on this. I thought about, could we do crystallography? But there are a few reasons why it's a bad target for crystallography. You've got this long, flexible protein called P150 glued, and that would make it hard to crystallize. And also, we can't actually make this recombinantly. So we're quite limited in how much protein we can make. We have to make it from pig's brains. We go to the butcher every Tuesday, get fresh brains, and we can get about 0.5 milligram of protein out of that prep. But that's then a perfect amount for Cryo-EM. And this person you may know, this is Richard Henderson. He won the Nobel Prize this year together with Jacques Dubochet and Joachim Frank, who will be talking at this meeting. And one of the things Richard had been pushing was the development of these new detectors that allowed Cryo-EM to get to really high resolution, as good or better than crystallography. And I shared a lab with Richard, and I'd like to tell you that, in our detailed discussions, he persuaded me to do Cryo-EM. In fact, I was far too focused on crystallography at that time, and I didn't really listen to him. Later, other people did, and Cryo-EM started happening in the Institute. And so at that point, my lab got involved. And it really got involved by two people that joined my lab. Linus came first, and he was a student. And I told him to go off down the corridor and find someone to teach him Cryo-EM. And amazingly, he did, and he's become a real expert at all of these aspects. And then later, Kai joined my lab, and he was a postdoc, came from China, where he'd done a lot of EM. And he's somebody who likes working on software development, so he does a lot of the methodology behind some of the Cryo-EM software. So together they started doing this structural work. And I don't know how many of you know how Cryo-EM works, but this is the sort of raw data you get. This is a micrograph of individual dynactin particles in a very thin layer of vitreous ice. And the idea is that when you average these together, you can get really amazing detailed atomic information. You also, of course, have to be able to see it in all the different orientations. And if you can do that, then you can build up a three-dimensional structure. So, of course, it's never quite as easy as that. There are a lot of challenges, and we faced a number of these that are common to people doing this method. So one thing was our sample was quite thin, so it was a bit difficult to see. The contrast wasn't so good. It had a tendency to fall apart. So if we used very nice thin ice where the contrast was very good, that completely destroyed the dynactin particle. And then the thing that really killed us for a bit was that it's a preferred orientation. So when we first started doing this averaging and getting these class averages, you could see we got these images, and there's a lot of detail in there. You won't be able to see it on this screen, but they're all sort of squiggly lines, and that tells you that you can see the secondary structure elements within this dynactin molecule. But as you can see, we always saw the same view. And so we had to do a few different things to get over this. We used thicker ice, we used a carbon support layer on the grids, and we also modified the protein with a crosslinker, which changed its surface properties enough that it laid down on the grid differently. And when we did that, we were able to see all these different orientations of the molecule and able to get a proper 3D reconstruction from it. And so this is our map, and I've colored it according to the different subunits that we knew were from this model. And it was 4 angstroms overall. And what that means in cryo-EM terms, in the center of this filament, you're at about 3.5 angstroms. And that's really good. That's good enough to see individual beta sheets and nucleotides, and you can really build that structure and get an atomic model. Out about 5 or 6 angstroms, but that's still good enough to fit in helices and build the structure. Now, the one thing you can't see in this map is this long, flexible region. And we assumed that that would just be flopping around so much that it would be impossible to see. But the other really cool thing about EM is that you can trap some quite rare states. And it turned out that in about 10% of our particles that we looked at in ice, this long, flexible arm was actually docked against the side of the dynactin. And that was enough for us to be able to get a structure of this at about 12 angstroms. And this actually completely changed the way that we thought about this arm. Previously, they'd seen this first bit, which is about 24 nanometers, assumed that it was this end part of the protein that was sticking out, and that these blobs here were this cap-glide domain. But actually it became apparent that really it's this whole region, it's much longer than that. This whole region sticks out, and that I think has some importance for how dynein is regulated. Okay, so from all of these different maps, we were able to put together a 3D model of dynactin. And you can see that it's built of this filament that's made of these 2 protofilaments of the ARP1. So there's 5 copies in the top here, 3 copies in the bottom. And it really looks very much like an actin filament. You can superimpose it with the actin structure. Down here, there's actually a beta-actin. Now, there'd been a big debate in the dynactin field, I guess in the late 90s, about whether actin was there or not, or whether it was an artifact of purification. But our maps were sufficiently good that we could really see that that was actually an actin. At this end, you've got capping protein. This is the same protein that caps off actin in the cell and is important for lamellipodia formation. We think it binds a bit more tightly to the dynactin than it does to actin, and we can explain that. And then at this end of the molecule, you're actually also having to cap off and stabilize this filament. But in this case, you're using another actin-related protein called ARP11, which does the job. And then there are a few other things that bind. But my favorite structure is this shoulder up the top. It's this amazing bundle of alpha helices. There's 8 different proteins in there. And when I play the movie, you'll see that it consists of these arms made of these long bundles of alpha helices that wrap over. Okay, so the first question we wanted to ask when we had this structure was, how do you get a filament, but one that's actually always the same length? And, you know, if you purify ARP1 on its own, you get variable length filaments. So there's some mechanism that really governs this. And we got a clue to that when we started playing with the threshold of the EM maps. And what perhaps you can see here is that there are these extra peptides coming out of the shoulder. And that will be more clear when I rotate it round and you can see it from the back view. And so what perhaps you can see is that these peptides are snaking out of the shoulder and are contacting all of those different ARP1 filaments. So they're acting like a little molecular ruler to determine the length. And so what happens, we think, in assembly is something like this. These filaments pick up the ARP1s, bring them to a high concentration to nucleate them to form a filament, and then that forms. We think actin binds perhaps just because there's so much of it in the cell. It's capped off with this ARP11. The other end is capped off with capping protein, and then the rest of the subunits form this structure. So this is pretty speculative, obviously, but I think probably it will be some sort of mechanism like that. It doesn't. I think that the concentration of ARP1 is probably very low in the cell. So there isn't enough of it really to even nucleate a filament unless it's bound to these peptides. So it's almost like you can only form the little ART1 filament when you gather it together. And that's enough to prevent the treadmilling. And then, of course, it's stabilized because there's lots of capping protein around and ART1. That's just my guess, but not a very good question. So next question we wanted to ask was, how does dynein bind to this thing? And to do that, we actually had a recombinant system for making dynein. We express all of the different heavy chain components off a single baculovirus, and we stick them together. And that's important for getting nice protein. We also use codon optimization a lot to make these genes, and that also turned out to give us 10-fold more protein than if we just used the native human genes. So we can make human dynein recombinantly, and the question is, you think you just mix it with dynactin and it would bind, but actually in vitro the affinities of dynein and dynactin were quite low. So what we needed was a trick to bring them together into a complex, and this is, I think, perhaps the adapter that Steve was talking to. This is a protein called BICD2, and this is work from the Atmanova and HOOK labs. And BICD2 is a long coiled coil. This end of it binds to the Rab6 protein, and that's connected to Golgi-derived vesicles. And at this end, you have the coiled coil that's sufficient to make a tight complex between dynein and dynactin. So we used our recombinant system. We chopped off the motor domain so we could just look at this complex, and we were able to get a structure of that, and that's shown here. So you see the coiled coil runs along the length of the a dynactin filament, and then the dynein tail here sits on top, and is the coiled coil is sandwiched in between. So we know from other work that the motor domains are going to be down here, and that makes sense because the C-terminus with the cargo binding region is up here, so you've got motors down one end and it's dragging the cargo along. And then the question is, how are you actually binding? And what you might be able to notice here is these two chains of dynein are actually arranged almost sort of parallel to each other, this structure and this structure. And they fit into the grooves on the dynactin filament. They actually sit into almost exactly the same site that myosin uses to bind to actin. And I think that's neat because it sort of explains why you need a dynactin, an actin-like cofactor. It basically matches the structure of dynein. And then the coiled coil, you can see as I play this movie, is really sandwiched in there, and it's supporting all of the interactions that dynein makes. So that's how you get this higher affinity structure. Okay, so what I've told you about the structural work, I think really changed at least my view of how dynein and dynactin worked. Previously, the thought was they were fairly loosely coupled complexes, and dynactin was the adapter that binds to cargo. But instead, I think what's happening is that the cargo adapters are here. They assemble then the dynein-dynactin machine, and it really comes together to make this quite tight complex. So what I'd like to do now in the next part of the talk is tell you a bit about how that machine is regulated. There's lots of different ways in which dynein is regulated in the cell, and the expert of that is much more Steve than I. There's things like LIS1 and Noodle, there's phosphorylation, there's lots of things going on. But what I want to focus on is the core mechanism of how you switch dynein on when you have one of these cargo adapters present. And this is the work of Kai again in my lab, and also Helen, who joined my lab as a technician. And I'd sort of assumed that she would help people with protein purifications and generally do, lots of different projects. But it turned out that she was really very determined to have her own project and work closely with someone. And so she and Kai together did all of the biochemistry and structural biology on this. And so the regulation then I'm talking about is this. If you look at mammalian dynein on its own, it doesn't move that much on microtubules, at least in an in vitro system. Whereas when you add dynactin to it, single dynein molecules can now start moving much longer distances processively. So I can show you some of the data behind that. This is one of the chimegraphs, distance along the microtubule here against time. And you can see that dynein on its own either stays in the same position or sort of diffuses around or takes some short movements. But contrast that to when you form this complex, and now you're getting these long-distance movements along the microtubule. So the question we wanted to address then was, what is it about dynein that means it doesn't go so well, and what happens when you add dynactin to switch it on? When we were starting to think about this, a paper was published by this Torisawa et al. And what they suggested was this sort of idea of how this could work. They looked at just dynein on its own, and they saw that sometimes the motor domains were stacked together, and other times they were separated. And so what they suggested is that the stacked form is inhibited, it can't move, and then it can separate, it can perhaps take some steps, but then it goes back to the stacked form, so it can't move. So it's a sort of auto-inhibition of dynein. And we wanted to test that, and we thought the first thing we should do is get the structure of this, as best structure as we could of this stacked phi-particle. And this was quite a challenge, actually, because although Cryo-EM is very good with flexible structures, this is really quite flexible. The motor domains are moving and the tail is moving. But Kai managed to do all sorts of amazing tricks with EM. He was able to get a 15 angstrom structure, and that's shown in this grey outline here. That showed you the overall sort of conformation of the structure. And then he could focus in on the tail and get a structure to 8, and the motor domain to about 4. And if I play this movie, you can see now that these heavy chains are wrapping around each other, are binding these different accessory chains of which we had structures known or good models. And then the light chains are in orange there at the back. And although in this representation it's not so easy to see, this is really a good 8 angstrom map, so we can actually place helices and build a sort of rough model of this tail region. And so here you can see it's made of this bundle these bundles of alpha helices that wrap around each other. It's held together in 3 places, at the top by this dimerization domain, down in the middle here by this intermediate chain. It has these long peptides that come out of it, and they're held together by this LC7. And then down the bottom here, there's a contact that's very specific to this phi-particle, actually between the 2 heavy chains. So this, I mean, for a dynein person, is fascinating to see this in 3D for the first time. But in some ways, the bit that we'd wanted to get to was this motor domain here. And because that's 3.8, you can perhaps just begin to make out, you can see the individual helices here. That's good enough resolution to really build an atomic model. And so we were able to do that. You've got, in dynein, you've got this ring of AAAs, which form the motor. You've got the stalk with the microtubule binding domain. And then this linker in purple is the thing that moves in order to drive that movement along the microtubule. And as I play the movie, you can see that they stack together with their linker domains on the inside, the stalks crossed, and you know this is how it's held in a weak inhibited form. But what this allows us to do then is to see the interface between these 2 motor domains and make mutations to separate it. And so what we can do now is actually test that model, because we can force everything into the open form. And the prediction would be that it should be able to walk along the microtubule on its own, no need to put dynactin in there. So first thing we did was to test that we had actually been able to open up the dynein. So we used negative stain EM. We collected lots of particles, averaged them together. And when you do that for wild-type dynein, you see that most of the particles have this stacked head conformation, and a small percentage have an open form. So there's an equilibrium between the 2. Whereas in our mutant, we've then really managed to open up all the motor domains and completely separate them. We don't see any of this phi form. So, question is, do you get a dynein that moves on its own? And to our great surprise, you don't. So this is wild-type dynein. It's either stationary or moving short distances. The mutant really doesn't activate it. And the first thing you could say is, well, maybe you've inactivated this dynein. But we can test that. We can do the control, and we can add in dynactin. And what you see then is that you've got beautiful movement in both cases. So this motor's absolutely fine. Was the wild type. The speeds was the same. There's a lot more activity here, and I'll get to that in a minute, but the speeds were basically the same between the two. Yeah. Yeah, so this is a chymograph, and each of these little squiggles is a binding event. So it binds to microtubules fine, it behaves just like the wild type. It's just, it can't walk on its own. So the point is, this is inhibited, but so's this form. So what this did is it changed the way we thought about this. What it means is that this form is also inhibited, and something is happening on dynactin binding to activate it. And we can look at what that is by going back to Cryo-EM, or EM in general. Now, because this open form is even more flexible, you're not going to be able to get a high-resolution structure of that. But we can use negative stain, we can collect lots of images, and we can actually see the flexibility. It's really cool. What I'm showing here is one of the class averages, and I'm going to play you a movie where I show you all of the different class averages, one after each other. And you can see the flexibility of the structure. The motor domains are moving around. But what I hope you can notice is that actually these motor domains are always pointing towards each other. That's shown in this cartoon here. In the sort of the raw images, you can see the stalks coming out, but you'll be able to make out this bend of the linker region. And so what this means is that even though the legs are apart, they're still sort of pointing towards each other. And that's not good if you want to try and move along a microtubule. So the question is, what happens when dynactin binds? And to do that, we actually looked at a cryo sample. And the reason we did that is in a cryo sample, you can actually subtract out certain parts of the structure. So we could subtract out the dynactin and just look at the dynein that was left. And when I play this movie, you'll see what's happened now is that the motor domains have all been flipped round. So they're now parallel to each other. And that's important because that matches the microtubule lattice. And so what we think is happening is that you've got these two inhibited forms, but it's only when dynactin binds that induces these long-range rearrangements that match it up for the microtubule and allow it to keep moving. So that was nice. The one question you may be wondering, full-length recombinant dynein. All-tailed, yeah, absolutely. Full-length, yeah. That's a good question, actually. I don't know, possibly. I mean, in the case of for phi to bind, yes, presumably only one's bound. Yes, I think you're probably right. Maybe one. But this is probably an equilibrium, right? We sometimes see them parallel. It's just very rarely. Whereas in this case, we see them matched up. So basically, sometimes this may be able to bind with two heads, but it's unfavorable. So it wants to be in this form. Hello at the back, sorry. Well, exactly. So let's get to that. That's the next bit. Was there another? But yes, that's a real important question. Yeah, no. What it is we're collecting lots of images of the complex, and then we're doing 2D averages of them. And they're all slightly different because there's lots of movement, but we can class them into ones that are most similar. So this is just randomly distributed things, but we are gathering the particles together that are most similar in order to be able to get a whole load of these images, each of which represents dynein. Did that make? Yeah. Yeah, so we don't know anything about the time. We know that there's a distribution in the whole population, and then we've frozen it in ice, and we've trapped that distribution. And then we're just imaging that distribution. Yeah. It's in a thin layer of ice. So, yeah, inside the ice, exactly. Whereas this one is on a carbon surface with negative stain on. Sorry, one. Yeah. We haven't, but another group have, and I'll show you that image very shortly in the next part of the talk. But yes, the people now can. It's very cool. Okay, so to your question, why, if they're both inhibited, why bother having both inhibited forms? To get at that, we went back to our mutant dynein and we asked how it was different. And we actually asked whether it could make dynein-dynactin complexes differently. And the answer is it can. And so what I'm showing you here is a gel filtration trace, which separates based on size. So this is the BICD sample. So we mix together all three components, BICD, dynactin, and dynein. And in the wild type case, which is in black, you get a small peak of that complex formation. But then when you make the mutant, which is in red, you have almost no dynein left because all of it has been shifted over into this dynein-dynactin complex. So that means that this phi-particle is much, the phi-particle can't bind dynactin, and when you open it up, you get something that really binds better. Is that important in cells? So to get at that, we worked with Alex Bird and his postdoc, and they can express the whole dynein heavy chain gene on a bacterial artificial chromosome, and it's GFP tagged, and they also made for us mutations that we made for our in vitro work. And they made two different clones, and you can see that both of these express at roughly similar levels, and the levels are quite low, so they don't disturb the cell too much. And then Alex looked at what's happening during mitosis, for example, and here are the spindles from these different cell types. And in the wild type case, you see the dynein throughout the spindle. That's very similar as if you do an antibody staining against dynein. But then in these mutant cases, the dynein really accumulates at the minus ends, suggesting it's been activated and it's all going to the minus ends. And what's even better is that the P150 also accumulates there. So that suggests in the cell, if you can't form the phi-particle, you get mislocalization of the dynein. And actually, Alex also saw a mitotic defect in there. We think this may even be important in humans. There are some human patients who have mutations at that phi interface. So what I've shown you then is that even at the core dynein-dynactin machinery level, you've got several steps of regulation. You've got this phi-particle, that somehow is regulated, it comes apart, and only then when you have the cargo present, which is that adapter protein, can you get the whole complex and it walk off along microtubules. Right, yes. No, and we've thought a lot about it. It's an important question. So one possibility is that when you attach to the beads, you're somehow, because you're attaching presumably by some part of the tail, perhaps you're disrupting that phi-particle, because the phi-particle itself is stabilizing, the tail is stabilizing that phi-particle, so you might somehow change that. The other possibility is that when dyneins are together, that the phi-particle is always coming apart, so then one attaches and another attaches, and they sort of feed back off each other. Do you see what I mean? So if you have a single molecule and it comes off the microtubule, it's gone, whereas if you have multiple, it has a chance to come off and come back on again. So no, you're doing single. So in that case, no, for that case, sorry, you're absolutely right. I suspect it's something to do with the attachment. It's doing something similar to the dynactin. That would be my guess. It's the one sort of bugbear we have with this is how do we rationalize that with the beads? Right, so what I want to do then in the last little bit of the talk is to take you through another question. So far I've shown you this BICD2 adapter, but the question that I mentioned at the beginning is how do different cargoes bind? And I just mentioned that this work is from Linus again and a new postdoc in the lab, Clinton. And so work, particularly from Richard McKenney, who was in Ron Vale's lab, suggested that there are a whole load of these different adapter proteins. So BICD2 is one of a number. And these are a list of different candidates. And the ones I've shown here in red are the ones that Rick has actually shown can do what BICD does and activate that long-distance movement of dynein. So are there any common features of these? So surprisingly, there isn't really much sequence conservation between them. But what a lot of them have are these stretches of long coiled coils, about 200 amino acids long. And so what we wanted to ask is, are these interacting with dynein-dynactin in the same way, and is there any sort of interesting variations? And so we focused on two of them. BICDR also binds to Golgi vesicles, but it's used in different developmental stages. It's used in early neuron development to change the properties of those vesicles. And Hook3 is involved with transporting endosomes around the cell. And so we solved structures of these at about 8 angstroms, and I'm just going to show you the model of the dynactin and how the coil-coil region of these adapters runs. And so in BICD2, which is in orange, I showed you before. And the first thing to notice is that all of them run along the dynactin filament. So that seems to be a common feature. But when you look more closely, they're actually all slightly different. So you'll see the BICDR interacts with the different point on this pointed end complex. There are differences down here. They actually have a different twist to some of these coils. So there's really quite a lot of flexibility in terms of these adapters and how they work. But in some ways, the most surprising thing came when we looked at the dynein part of these structures. Because now, instead of just one dynein, which we'd seen in the case of BICD2, these two adapters now recruit two dyneins parallel to each other on the filament. And I was very excited about this because there was quite a lot of evidence, there were some hints in the literature that both of these adapters move maybe twice as fast as BICD2. And so sort of naively you think, okay, well, two motors going twice as fast. But if you think about it, you tie two marathon runners together and they're not going to run a marathon twice as fast. So it was a sort of conundrum. The model I like to come up with, I called the tethered toddler model. So the idea is if you have a young child, a toddler, and you want to get them to walk somewhere, they're going to wander off and look at flowers or play with their toys or something. Whereas perhaps if you tied two of them together, one would pull the other along and they would somehow manage to get there faster. And there's some suggestions in the literature this might be a good model for dynein, because it was known from some yeast dynein work that dynein takes a lot of backwards and forward steps and sideways steps. So the idea would be, in the case of BICD2, where you only have one motor, you're taking this very roundabout walk. Whereas BICDR, you're bringing them together and they're all going much faster. So this was turning into a really nice story. And then, of course, things got a lot more complicated. Because I went to biophysics and I started chatting to Daniel Grotjan and Saikat Chowdhury. Saikat, I think perhaps some of you may know. And they're from Gabe Lander's lab, and they'd been doing this beautiful tomography work, and they'd also seen these two dynein complexes. This answers your question about seeing them actually on the microtubule. And the thing is, though, that they've got this BICD2 adapter in there. This is the one that we said only binds to one dynein. So, your first response in a case like this is, they're wrong. Your second response is, we may be wrong. And then after that, you sit down, you do some experiments, and of course it turns out that actually we're both right. It's all fine. So the experiment that we did was to actually now collect negative stain data rather than a Cryo-EM data on a purified complex. We just mixed all the components, looked by negative stain, and saw what type of complexes are formed. And what we found is, yes, indeed, you can get these two dyneins bound, but it's actually only a fairly low percentage. It's 20% of the molecules, whereas 80% have one bound. And you compare that with these other two adapters, and in this case, pretty much every molecule you have has two bound. So it's not quite as simple as 1 versus 2, but there's a shift in the distribution from mainly 1 to mainly 2. So the question is that enough to change the speed? And so get at that, we first measured the speed in our in vitro system. And you can perhaps already see from these chymographs that there are a lot more of these slower runs in the case of BICD2, whereas the distribution has shifted. So you've got the same distance over less time, lots of faster runs in the case of these other two adapters. And when we quantify this, we don't have quite a twofold, but there's a clear, at least 1.5-fold difference between these. So it's definitely getting faster. So to get at the question of whether two were required, we did a single molecule assay where we labeled dynein, full-length dynein in one color, and then we labeled one of our tail constructs in a different color. And so now you know that if you see both colors on a moving spot, you've got a one-headed dynein complex. And if you see only one color, then you've got a two-headed complex. And we looked at the distribution, and we saw absolutely this speed was reduced back to about this same level. So this really suggests to me that it's the presence of this second dynein which is helping to increase the speed. There's also an increase in force, which is what you'd expect, and I can show you that if you're interested. Okay, so what I want to do next is to ask the question of how do you get these two dyneins bound? And the nice thing about, and so the thing we had to do then was to see if we could get this complex to as high a resolution as possible. Yes. We haven't actually quantified that yet, but we have to. And yes, I suspect it will make it more precessive. That would be our suggestion. We can't measure that. That would be nice to look at, but with this assay, we can't get that resolution. Yes, no, absolutely. I suspect it would be the same, but it's something we definitely should look at. Okay, so how do you get to high resolution? So we had to collect a lot more data. So previously, we got 1 day's data collection. We managed to beg, borrow, and steal 11 days worth of microscope time, which is incredibly valuable at the moment. We collected 7 images on each grid hole, compared with typically just 1 or 2. We really packed in the samples. So remember, on that first image I showed you that all the particles were nicely distributed. Here, you're really packing them in as you can get them. And so we had about 1 million particles, and that should have been enough to get us high resolution. And sure enough, from this, we were able to get a 3.5 angstrom map, and that was beautiful, a great improvement on our previous one. But the thing you'll see here, this is a resolution map. So blue is the high resolution and red is the really low resolution. And the bit that we cared about most, the dynein, you'll see is sort of much lower resolution. But you can do these tricks in EM. You can subtract out the dynactin signal and just focus on the bit of the molecule you think is sort of most stable. And so Linus did this and was able to define the bit of the molecule that was this most stable chunk. And he split the dynein into 2 bits and was able to get high resolution of both ends. And the real thing that he did was just keep on playing with the region he was going to include in order to get a really high resolution structure. And so here you can see some of the maps, and those of you who have seen crystal maps will know that this is getting there in terms of resolution. So for the first time, we can build the structure of the dynein tail. You've got this N-terminal dimerization domain, you've got all those helical bundles, that mouse mutation I showed you is in this loop here, and then it goes up and binds this intermediate chain and light intermediate chain. We could build this structure, and we could model in this structure based on a previous known crystal structure. And so we can look at the interactions of those chains. Intermediate chain is a WD-40. It binds to the heavy chain using this loop, this helix, which dips down into the central sort of a pore of the WD-40. That's actually a classic WD-40 interaction motif. But the most interesting of these interactions was the light intermediate chain, because this actually makes only fairly weak interactions between the globular part and the heavy chain. But it has these long sort of tent ropes that come out and hold it tightly onto the heavy chain structure. And at the end here, there are these helices which are intercalated into this heavy chain structure. So we were able to put together then a map of the whole complex using our 3.5 angstrom structure. We built one of these chains and we docked into the other positions. We could build in parts of the adapter here and the dynein, and I'm just showing the dynein in a surface resolution. So how is it recruited? So the first point is that this dynein A, which is in blue, is sitting in these grooves. That's binding exactly in the same way that I showed you for that previous BICD structure. This dynein actually one of them sits in the groove between two ART1s, but the other one actually sits between an ART1 and a capping protein, so it uses this different cleft structure. And that allows it to bend round and positions it properly. It connects, of course, to the adapter. It uses these helical bundles 2 to connect to the adapter and helical bundle 5 for this dynein A. Dynein B, though, is a bit different. Only one of the two chains actually binds to the adapter. And although it uses the same areas of the chain to bind it, this interaction here is actually mediated not by a direct connection to the adapter, but by some extra density. And we have a suspicion that this is the light intermediate chain, the very C-terminal portion of that. But the biggest surprise to me was just how many interactions there were by the two dynein molecules. So this dynactin is arraying these things, but then they're binding to each other. So here is the A and the B chain next to each other. And each of these spheres here represents an interaction. And so they're absolutely tightly held together. And I think that's probably very important for that orientation of the motor domains that's so important for this increased activity. So I'll end there. Just to summarize, I've told you that dynein-dynactin is a machine that comes together in the presence of these cargo adapters. We think one of the activation processes is by dynactin aligning these motor domains. And then I've told you that there are a whole load of different adapters, and some of them can recruit multiple dyneins for faster speeds. So with that, I'd just like to thank again my group, particularly Linus, Kai, Helen, and Clinton, Alex and Arno in particular, my other collaborators who work on this, and the EM people at the LMB who have been so helpful in everything that we've done. And I'd like to thank you all very much for your attention.
On the microtubule, which is opposite to some charge, maybe on that extension of the dynactin, right, which may help in tethering. Is there any such an evidence of that? Is that redundant?
So, I mean, this may be a question Steve can help me with. I think that the idea is that that interaction is not necessary for dynein's function in cells. I think the picture is coming that the part of the molecule you're referring to, this basic domain and cap-glide domain, are much more important for targeting dynein and dynactin to the tips of the plus-end tips of microtubules, and that they're not absolutely required for the movement, the processive movement.
Doesn't have a motor activity. So in that sense, then how does it control the affinity for the track?
So dynactin is controlling the affinity for the track by putting dynein into the correct conformation to bind it. It's not directly contacting, we think. And there is some evidence for that. For example, even in these assays, the dynactin we're using is actually mainly a P135 isoform, which lacks at least the cap-glide domain. And so it doesn't have that interaction site, and yet we see quite a lot of movement. And there's other work from Rick McKenney which addresses that.
Certainly I'm comfortable with that. We had published a paper, sort of a biophysical analysis, that suggested that when you had dynein with dynactin, not with BICD, that the lateral motion was decreased. So that, I was wondering whether or not, you know, there had been some work a while ago by someone in Japan, and I've never seen it published, but it suggested that if you had lateral interactions between the two heads, they could affect the enzymatic activity of the heads. And so I'm wondering whether or not when you're having these multiple dyneins that are together, rather than sort of a model where they're wandering less, which I kind of doubt, because I think that, in our hands, it didn't look like it was wandering that much more than kinesin when you had the dynactin. I mean, it was a little bit more, but it seems more likely to me that those interactions that you're seeing, if they could be between sort of the lateral, heads, you could have lateral gating of enzymatic activity, which could easily give you an increase in speed.
Absolutely. And I mean, Gabe's structure is cool because they see contacts between the heads here. So yes, that could be affecting it. I still quite like the wandering experiment. We've tried to do some high resolution trapping, and certainly our BICDR traces look straighter than our BICD traces. I think our BICD traces are too noisy to really be confident about that. But I think, that's the sort of question. It would be nice to revisit that with the sort of the whole system and these two adapters. But absolutely, some sort of enzymatic thing is a possibility. Oh yeah, LC8 is cool.
Is critical in dimerizing IC74. So
the LC8 and TC-TEX, perhaps I could go back to this model. So you've got the intermediate chain here, and then you've got the long N-terminal extension, and it's zippered together in 3 places by LC7, LC8, and TC-TEX, and we know that from the structural work from the Barber and Williams lab. But what's interesting is that we see some extra density, and we can't be completely sure what it was, but we see a little bit of a connection between the LC7, which we really can see, and this density here. So it looks to us as though these light chains are actually docking against one side of the heavy chain. So in this inhibited form, they're actually zippering down and helping to stabilize the structure a bit. We haven't. We've just actually made those constructs recently. It's slightly more complicated to make those truncations, but we've now got some nice clean truncations that lack each one of these chains. So we hope to be able to do that. Yes, and I think in the dynein itself, then you can imagine that there's a connection from here to here, and then when the dynein opens up, I think this is then very flexible, and Gabe Lander has some evidence to suggest that.
So when you say adapter is helping recruiting more dynein, that's what, is that I probably did not get, so the second dynein, which is coming, so you say that adapter is helping to recruit that?
Yes, absolutely.
So what exactly, so adapter is driving, helping some conformational change in one dynein to bring the other dynein, or what?
No, it really...
And the spatial arrangement of the 2 dynein you are showing, so...
Yeah, no, it really just adds, it adds interactions. So let's see if I can just get to... that. Sorry, let me play the movie. It's probably the best thing. So the adapter here is actually interacting with both the dynactin and to the dynein. So it's as though it's adding extra support. So the interaction of the dynein with the filament is quite weak on its own, but then when the adapter's there, it's adding enough extra contacts to stabilize it. And there are slight differences between the BICD and the BICDR, for example, which controls the position of this coiled coil and allows it to either get 1 or 2.
Meaning that, are there this is one of the conformation possibly, so is there a energetics or free energy analysis to show that what are the possible modes of in presence of...
There's no free energy analysis, and I think that would be helpful. What we've got is we've got some negative stain reconstructions at quite high resolution of the BICD, which recruits either 1 or 2. And what we see is that the position of this coil actually shifts. So let me go back to this first slide. So you see here the position of this coil shifts between the 2 structures. So in the case of BICD2, when you've got 2 dyneins bound, this orange has shifted down to be in exactly the same position as the BICDR. So there looks like there's a conformational dynamics of this on the dynactin structure.
I see.
Sorry, is that?
My question is related to what Probal asked. So, is there a number of dynactin, or essentially, is there a distribution that the number of dyneins sort of come together and act essentially as a cluster?
Yeah, so one thing to be said is, in the cell, it could be much more complicated. There could be a lot of these adapters, and there could be multiple dyneins involved. But in this case, in the case of BICD2, I think there is an equilibrium between the two, and maybe other factors in the cell can shift that equilibrium one way or the other. In the case of these other adapters, it looks like 2 is really the dominant form. It looks like it always gets 2.
Okay, thank you.
Okay. Oh, well.
You mentioned in passing that the force exerted is larger when you have 2 dyneins in these complexes. How do you determine that?
Yeah, so it was done by optical trap. Time up. That was 55 minutes, so we're okay. Yeah, we did an optical trap. We worked with Ahmet Yildiz, who's in Berkeley, and he attaches the optical trap to the adapter and then can recruit it. He's also attached it to the dynein and done the controls. So this in his hands is kinesin. This is BICD2, which was about just below 4. And then BICDR and HOOK are higher. BICDR is the highest force. Weirdly, these 2 are different forces, and we don't quite understand why that is. Thank you. Great, thank you very much.